And I was reading the book just the other night about how we can make sure that we serve all students at the university. And one of the statistics that really jumped at me was that very few schools nationwide offer internships or research experience as required. And most of them are optional. And not many schools offer those either at all. In some schools, it's really hard to get into research or internship. So I guess from that perspective, our school is somewhat unique, right? Because in chemistry, for example, we require capstone experience. And in biology, there's group capstone experience, if I remember it correctly.
It's only required for the DS program, but we do offer a lot of options to take it, as an optional research course.
All right. That's pretty much it. I think the advantage for introduction, the advantage of having student-faculty collaborations is multi-level, and one of that is inspiration for studying, hopefully, and another is the buildup of one's confidence in ability to handle the future, let's call it this way, right? So I don't know, I would like to go around the table and engage first students and see if you can maybe first tell us what you're working on, and then the follow-up question will follow later. So like, do you see an actual advantage of that or not, or an alternative? So what feedback do you have for us as mentors so that we could do it in a way that would be more beneficial? Right, so I don't know, we have to start somewhere, right? So I don't know, Liam, how do you feel starting there on the top?
Yeah, I can go ahead.
So go ahead.
So my name is Liam Tovey. I am working with Dr. Haddadi, and we are trying to perfect a method of dispersive liquid-liquid microextraction for diphenhydramine, or DPH for short, which is a common date rape drug. To do this, we're making indirect solutions of aqueous solutions of DPH with ultra pure water and butanol, and then we spike it with 13-14 toluene acetonitrile solution. That's so when we vortex it, it will separate into two layers, and then when we freeze it using a liquid nitrogen and acetone bath, the bottom layer freezes so that we can easily extract the top layer, which is where the DPH actually is, with this particular 1340 toluene acetonitrile solution. So then once we get to the top layer, we evaporate it and then using nitrogen gas, and then reconstitute it with methanol just to get it back into an aqueous solution. And then we also make just the DPH and methanol for a direct sample, just so that we can later analyze our recovery rate. And to do that, we use gas chromatography, and then we look at the peaks for the indirect solutions and compare them to the direct solutions to see what the difference in peaks. If there's any difference, it shows us how good our recovery rate was.
Liam, if I may interrupt just for a second before we get bogged down in details. Why are you doing this project? What's the overall purpose for it? Because not everybody here is a chemist, right? So we want to be a little bit more introductory about it, maybe.
Yeah, so so with diphenhydramine, it's a very easy, so it's dissolved in aqueous solutions, obviously. So when we take a sample, it will be metabolized over time. And so depending on when we take the sample, it could be in very low concentrations. So that when we take the solution, if it's in parts per million or maybe even parts per billion after maybe a few days, we can still detect it by using this extraction method.
Okay, great. Thank you.
No
What
problem.
benefits do you see to yourself from having done this research?
So I want to be a forensic scientist when I leave for college. So this is completely up my alley. I really want to have that experience with maybe different extraction methods for drugs, and also to have this experience of having a problem, trying to fix it, and also to refine my methods. It's also very helpful for the forensic field.
All right, great. Thank you. Do we have any students there on the? Not at the moment. Would you please start the discussion here around the table?
Okay, yeah. Hi everyone, my name is Valeria Torello. I'm working with Dr. Yulia Artemenko on to better understand the mechanisms that are involved in a directed cell migration, specifically in response to mechanical stimuli. The understanding of the mechanisms is important because metastatic cancer cells move, immune cells move. So if we understand how they move and what mechanisms are involved, we can, specifically for cancer, we can have other ways of fighting cancer. And to determine how the cell senses mechanical stimuli, we plan on disrupting the sugar coating or the glycocalyx of the cells, because we hypothesize that it's involved in the sensation of mechanical stimulus, such as shear flow. And basically in the lab, we will be transforming the cells with a fluorescent protein sensor that senses polymerized filaments of the cytoskeleton that are activated in the sensation, and then perform adhesion and migration assays.
Very interesting.
See the effect of the treatment of the cells.
Sounds good. And the follow-up, the second part of the question, like how do you see yourself as a young scientist within this role?
Could you elaborate?
Well, do you see, yourself growing in this process? Where are you at this process? Is that beginning? Is it like how do you see this experience transforming you in any way, if at all?
I've been working in the lab for the past semester, and I've learned many techniques like the different assays that we did, how to use the microscope, and beyond college, as of right now, I plan on doing research. So any lab experience that I can get, and specifically cell biology, is interesting to me, and molecular stuff is interesting.
Good. So just true hands-on experience, right,
Yes.
in a wet lab?
Yeah.
Sure. Sounds great.
Hello, everyone. My name is Quinn Jones. I'm a junior, and I've been working with Dr. Yulia Artemenko for the past 3 or 4 months. I'm also studying directed cell migration, and my project has to do with a genetic screen. Basically, we have a protein that negatively regulates directed cell migration, and I am running a genetic screen to kind of identify genetic relationships between this protein and novel partners of this protein. And that's hopefully going to help us better understand the specific mechanisms that allow this protein, or this kinase, to regulate cellular adhesion and subsequently cellular movement. It's only been a few months, but so far the whole experience has been tremendously beneficial for a number of reasons. I'm not 100% certain on what I want to do with my career yet. I know something in the health sciences, but just generally speaking, My critical thinking abilities have been really challenged these past few months in a way that has helped me grow a lot as a researcher, as a learner, and as a worker in general. I've gotten a lot more comfortable with like scientific language and vernacular, and I think that's going to be a tremendous help down the road, no matter what I do in the science fields. Similar to Valeria, I've gotten accustomed to certain procedures and equipment that I'm going to see in the lab down the road that I think is going to be very helpful for whatever career I decide. And yeah, ultimately, like Kesta said at the beginning, my confidence in general as a scientist has grown exponentially in a short amount of time. So I still have 2 more years left. So I think by the end of 2 years of research, this is going to be, I'll be a fully fledged scientist, so to speak. So
That's awesome. That sounds
it's
great.
definitely been a very, very enriching experience, and I'm very grateful for the opportunity. So
I know I cry.
All right, Vikrant, you be next.
So my name is Vikram, and I recently graduated with BS in biochemistry. So I've been involved in research with Dr. Kestis for the last 3.5 years, and I worked on multiple projects, like 6 projects I worked on. I started working on CRISPR, and then eventually, we were trying to develop a teaching laboratory for CRISPR for biochemistry students, but somebody else published a similar thing, so we abandoned that, and then we had to move on to. Then I worked on extraction of creatinine in urine and its correlation to urinary metals in 300 children in Syracuse area. So we're about to publish that. So that's one of the I feel
Productive efforts.
productive efforts that I had. And then we moved on, then we worked on RT-PCR for COVID-19 to detect the limitations of RT-PCR while detecting COVID-19. And we developed a teaching laboratory for biochemistry students, and we have been running that lab for the last 3 semesters, and we're about to publish that too. And now I worked on measuring the physiological stress that is cortisol and its correlation to different diseases. And we have worked with various collaborators. And the goal of all the research is that I want to go to grad school to study further about metabolism and aging. So I feel like I had a hands-on experience for 3.5 years, and I feel myself educated and skilled enough so I can, take on the further advantages of grad school, and I feel myself prepared enough. So that's it.
Great, thank you.
All right, I think I'm in camera. I am Devin Seal. I'm going to be a junior starting next semester. And I think of everyone here, I am the newest one. This is day 10 for me. So I've just started. I'm working with Dr. Kestis, and I've been really enjoying it so far. I've been primarily working with the Cortisol. We do stress research for the purpose of, the main reason that I've been told that we kind of do this is that a lot of times for stress, you go and you get people and people question them and ask them, are you been stressed? How have you been feeling? For people with acute stressors, but some people will be very open about their stress and some people will be very closed. So we can do research primarily with hair to see the actual chemical amount of stress that they're experiencing, which is probably a lot more accurate than just asking someone a question. And the way we find that is that we've been doing assays to sort of find exactly how much is in that hair sample. And I've just started with that, learning the procedures, learning how to use the instruments. And I just yesterday did my second quadruplicate set of that. And so hopefully we'll see some good data from that, but I'm really enjoying it so far. I'm learning a lot more about how to use a lot of the different instruments. I've learned a lot more about pipetting. Just yesterday, I used a multi-channel pipette for the first time. So pretty simple stuff, but it's fresh to me. And I'm glad to be learning about it because I think obviously that experience is going to be very, very helpful in pretty much anything that I try to do. So I'm really happy to be given that opportunity to do that by Dr. Kestis.
All right, so we covered all the students here. I don't know what faculty want to say and how to frame it. We'll go in the backwards direction then, okay? So we'll start here and I guess, at least as far as my work is concerned, what I do, I emphasize proper controls and redoing experiments multiple times before we're confident, so that we don't convince ourselves in something that's not there, right? Just because we want to see it may or not be there. And we typically do that same experiment by 2 different students, so that we're like solid on that. Other than that, it's a lot of fun in the lab, teaching basic stuff. But then when we put it together and it gets a bigger story, that's the most important part, I guess. And I just enjoy it. I just enjoy the lab work. Somebody asked me sometimes a few months back, how do you encourage faculty to do research? I was like, you can facilitate it, but they either want to do that or they don't, right? So people who are here in this room and on the Zoom, I know that we're preaching here to the choir because we're the ones who actually, care and facilitate that. So I don't know. I'll just pass the word to the right.
Great, yeah, so I'm Dave Dunn, I'm in biological sciences. I think I know most of you here. At a very high level, I guess what I would have to add to this is that I'm coming at my research endeavor as a faculty member here kind of from 2 different directions. One is I'm interested in moving forward my personal interests, my research agenda. There's things that I want to study, there's things that I'm interested in, and that's a major goal, frankly. It's a big part of what I want to do. But the second thing, which is usually goes hand in hand with the first, but not always, is that my goal is to advance the careers, and as you students have described, help them gain experience, to gain abilities with techniques or with machines or with different concepts that will advance their employability or advance their ability to be accepted into good graduate school programs or professional school programs. And so, sometimes it's a balancing act between, I really got to get this data that would be so much faster if I just did it myself, versus, hey, let's hold this student's hand who's only been in the lab for a month and help them to do it right. And so, sometimes there's a little bit of give and take between the 2 priorities there. But I guess in a certain sense, I've kind of tried to, focus more on the students, and I think I've had some pretty good success doing that. I'm very pleased with many of the students who have come through my laboratory. It has been a joy. I received a phone call just a couple of months ago from one of my former students who graduated a few years ago saying, hey, I really appreciate that you gave me the opportunity to, I have a small colony of laboratory mice. The mouse is a very important laboratory model. He says, hey, I'm glad that I got to do all that hard work that I hated at the time, shoveling all of the, feces out of the cages and doing all this. It smelled bad. It wasn't very fun. But he's now working for Taconic, which is one of the largest suppliers of laboratory mice in the world. And he said, we need more people like me. Taconic's looking to hire. Do you have anybody else coming to the right? And so I personally felt really quite good about, having provided that student the experience and seeing that the value that he saw, even if it was after he graduated. And so, with research, I know that a lot of us, this whole room is sort of science heavy with the concentration of biological sciences and chemistry faculty, but to get into grad school, to get into medical school, vet school, etc., having research experience is super valuable. And I'm just very happy that I have the opportunity to be in a place where I can help facilitate that experience.
I just want to kind of echo the part about email. Getting one of those a year makes entire life worth living. Honestly, they don't come often, but when they come, once or twice a year, that is a really something that is a treasure. So, well.
Hi, everyone. I'm Yulia Artemenko. I'm an assistant professor in biological sciences. And yeah, just to echo what Dave was saying, I often say to the students that I realize we probably won't solve cancer, given we're in an undergrad institution. But I think that doing that research, we might make some advances and learn something, but more than anything, I think the opportunities that the students get to learn and just to go through the scientific process is invaluable to them. I mean, so many of them have that kind of upper hand in being able to get into grad schools and research positions afterwards, or med school, or whatever they might want to do afterwards. That I think it's definitely all worth it, even if we don't solve cancer, right? So I think it's definitely good for them. The other advantage I think that I found that's really, and I kind of experienced it back as an undergrad myself, is that doing these types of experiences teaches the students if they don't want to do it either, which I think is just as valuable for them to realize that, okay, research is just not for me, or maybe a different sort of program. I've had somebody go into grad school in urban planning, for example, after doing research in my lab. It still gave them the skills and the tools to be able to get into grad school successfully, but that's what they decided they wanted to do. Or somebody wants to go into vet school and decided that no, research is not for me, but this is still something that's going to help them get into vet school because it shows that they have the critical thinking, etc. So I think, yeah, I definitely value, not progress is great, and if we can advance science is great, but I think the opportunities for the students is kind of really the goal, given where we are at an undergrad institution. And it's fun to kind of see the students grow over the years, especially if they're staying for a few semesters.
I wanted to ask you a follow-up question. I know that 4 of your students just recently presented at the national conference. And I'm sure that they appreciated it, but what was your perspective from that event?
I know how exciting it is to be at one of these big conferences when you're an undergrad. I had an opportunity to be at one when I was doing research as an undergrad, and I think those conferences are overwhelming, even if you're a scientist, but as an undergrad, it's like a whole other world. I mean, they're huge, right? These big national conferences. So if I have an opportunity to send students to those, to be able to present their research and actually talk to scientists and other students in the field of cell biology, then I think it's really helpful for them. It's a different experience than presenting, let's say, at even at Quest or like a Rochester conference, where you have a lot of different fields of science. There's opportunity there's value to those experiences too, and being able to share your research with people who don't really understand what you're talking about. That's a whole skill in and of itself. But to talk to people who do understand it and actually ask questions, that's a tremendous experience for students, for sure. So yeah, if I can't send them, then I try to always do that.
All right, I think we covered everybody in the room here, and we have Matt and Shaku on line. Matt, before you disappear, why don't you go first and tell about your perspective on faculty-student collaboration?
Yeah, that sounds good. So when I work with undergraduate students, I sort of take a stepwise approach. And one of the first things that I try and do with students is sort of build the confidence of students. And that's because I think that oftentimes when undergraduate students are entering the realm of research, it's really intimidating. There's a lot of things that they don't know. Even if they've done organic chemistry in courses before, doing research in organic chemistry is dramatically different than what you're doing in a course. So first, with all of the students that come into my lab, I just sort of work with them to develop their confidence, make sure that they know that these are things that they can carry out. They can be successful doing research in this field. And then once they sort of gain that confidence, then we sort of move on to carrying out important techniques that they can do reproducibly, because as scientists, we know reproducibility is one of the most important things that one of the most important skills a student can have. So we sort of work on being able to not only carry out these techniques, but carry them out reproducibly in such a way that we can produce data that sort of pushes the field or pushes the project forward. And then once we sort of accomplish that, then we work towards introducing, or I work towards introducing the student to as many techniques as possible. So as an organic chemist, I should talk a little bit about what my field is. I'm a organic/polymer chemist, and we work on creating environmentally friendly, sustainable plastics. And so once the students sort of reach those first two tiers, then we can focus on introducing the techniques of not just organic synthesis, but polymer chemistry and materials chemistry, as well as we do a wide variety of analytical techniques. And so sort of building up the diversity of the students' knowledge base so that when they leave the research lab and move on to whatever their career is, they have as many different opportunities as possible. And not all students make it through all of these different steps. Sometimes it takes a little bit longer for a student to build their confidence, and that's okay. Each student sort of progresses at their own rate. But in general, that's sort of how I approach trying to prepare students through research for whatever their career goals are.
Thank you. Shuku?
Hi, everyone. It's Shuku Haddadi from chemistry. I really don't have much to add to what all of you mentioned. And I'm not really a good speaker, so I just try to about research. First of all, I wanted to thank the internal grants we get here. And I'm sure our students are very grateful for that as well. It gives them a great opportunity to put into practice what they learn in classrooms. And what I have noticed is that most of them, they don't have any idea of what research is, that they're even scared of it. They think it's something they can't do, or it's something very difficult. But when they come to the lab, and as Matt said, we break it down. We start with literature review and with simple experiments. So they get encouraged, they get into it, they become more enthusiastic about it. And at the end, they want to continue. And what actually motivates me to do research is mostly to see that enthusiasm and that the students actually want to do research. They want to go to a laboratory and work in such an environment, or they want to go to grad school and do more research. And that actually motivates me a lot. And I also try to mostly focus on teaching students how to do research. And of course, as a side effect, I get to publish as well. So that's a good thing. So that's all. And thank you all for all you shared. I agree with all of that as well. So I don't want to repeat what you just mentioned. If you have any questions, I would be happy to add to what I just said.
Right. Well, thank you. Thank you everyone for participating today. Just a couple of announcements. So we'll have a end of June event, somewhat similar to what we have right now, but there will be a generous and hopefully very good luncheon included with it.
We have chips.
And then in the fall, close to the mid of September, there's going to be a poster conference, and everybody is welcome to participate in that as well, and as a wrap up for the summer months. If you have any suggestions on how can we run programs on campus better, that would be, that feedback would be very appreciated. One thing that I know myself is that I would prefer to turnaround time for submitting orders and getting supplies at a better pace than it currently exists. And for those of you who do not know, many of you do, but for those of you who do not know, there have been a couple of improvements lately. So the total amount of money that is available for travel funds has increased to up to 500 per student. And the same thing for small grants. So if a student needs supplies, it's a 1.5, 2-page application, and you can get up to $500 for supplies for the project. So faculty, student mini grants and student travel grants have been I guess substantially improved. But we'll see how it goes, in the future. Any questions?
I have a question for other faculty mentors. What are your ways for motivating students that might be losing motivation? I'm not talking about these two, they're very, no issues with them at all. But, when you have a bunch of students, some tend to put in sort of more effort, more time into research than others. Just want to hear ideas, I guess.
Well, we'll start at the end of it. So Shiku, here is your chance.
So I'm not sure if I got the question right. Was it how we motivate students to spend more time in the lab?
Sure.
Yeah. I think encouraging them helps a lot, like helping them gain confidence and showing them what they know as opposed to what they don't actually know. That will motivate them a lot, because if you start with criticism and showing them all their, shortcomings, they're going to get discouraged. But if you start with their strength and show them how much they can do, that will encourage them, that more enthusiasm. Dr. Brown in our department, I really learned from him. He goes with excellent, good job. And I really like that because, it helps students build up confidence. And I try to use more of those because I don't think students realize how powerful they are, how much strength they have in them. If we don't mention that, they're new to this. And we take it for granted, we think they know. But they'll have all these questions. Did I do it right? Am I doing a good job in the lab? So telling them that I think helps. That's what I have realized, because I'm kind of, I can say, a bold person, don't show my expressions much, but I've learned that, just showing how you feel about what they're doing in the lab helps. And with my students, I think most of them are motivated at the first place, and I just try not to kill the motivation in them. So, so that's all I have to say.
Yeah, thank you. And I see that Steve is online. His camera was not on, so I kind of unintentionally skipped you, Steve. Would you like to chime in and tell us about your work and your work with students?
Yeah,
My
hi.
apologies.
Yeah, I mean, you want to just share with you working with them? And yeah, I mean, last
Tell about
summer I
your work.
worked with a student, and the summer before I worked with a student. And I know in our program, we encourage students to do independent studies and to do some research, but over the summer, boy, it's wonderful. It's more one-on-one interaction, and it's just a joy. And you give them some tasks to perform, and it's face-to-face conversations, and from day to day, and they're learning. And sometimes you can learn from the student. It works the other way. They have ideas, they generate ideas, and when that happens, it's wonderful. I mean, they're thinking outside the box possibly, or in a different way or different perspective, and that's just that's valuable when that happens. And usually it's what you guys usually work with seniors or juniors. Usually it's upper class. In my case, it's juniors, and they have quite a bit under their belt. They've learned quite a bit and know how to use many of the tools. I mean, for us, we have a tremendous amount of data that we need to process and do diagnostics on, but then go further with that and try to make sense of it all within the framework of our science and physics and everything else. But that's I mean, like I've heard all you talked about. You really enjoy doing the research. It's in you.
Do you have an answer for Julia's question?
Well, what's the question now?
How to encourage people students who lose enthusiasm?
Oh, well, again, you have to show them that, or they have the know-how already. I mean, you give them some task and there's tools that they can use. And in our case, maybe like with some writing a Python script to process data, or to think about, to do some diagnostics, compute some quantities, and try to make sense of what's happening in the atmosphere, whatever event you're studying, and it starts clicking for them. I mean, you want to be enthusiastic, and you want to show that to the students, because that will get them enthused too. If you have that in you, the student sees that and they appreciate it. And it works the other way too. You want to see the student be enthusiastic. I mean, is it the same for you guys? I don't want to ramble on here.
That's okay. Rebecca, would you like to chime in about your work and collaboration with students? And then we'll ask Michelle the same thing.
Oh, I'm happy to. I'm here, I think, as the audience member. And I have questions for all the panelists. I'm a cultural anthropologist, Rebecca Peters, and it's delightful to read your applications when you apply to the research grants, and delightful to hear you all speak on all of this. My question for you today, I think, is especially for the faculty mentors, but also for the students. How do you navigate the research interests, right, that are most closely aligned with the ones that are a little more divergent? And let me just say a few more things before we come back to that question. So as a cultural anthropologist, I work in Southern Africa. I work in Zambia, I work in Angola. Many of my materials are in other languages that are not English, and my students are very interested, but it is hard sometimes to carve out a section of my work that they can contribute to if they don't have the linguistic skills and the cultural background and so on. But I'm very interested in encouraging them on their interests, right? You can do anthropology anywhere. They can do local research projects. I'd love to help them with that. But as many of you have said, right, sometimes they don't know how yet to do research, right? The whole point is this apprenticeship approach, and how to apprentice students in my work when maybe they don't want to do stuff in Africa, maybe they haven't ever been there, maybe they don't speak Portuguese, right? There's just a lot of like logistical hurdles I struggle with. And then how to encourage them on their work, which is maybe more local and on different topics, right? So how in your work, each of you, have you carved out like the appropriate space for students, right, in your own work, and then encourage them on their own path that maybe isn't about your particular trajectory? Is that too hard? I'm sorry.
I can start if you want. I'll just say that I'm not actually very good at doing that, because, and the reason why is I find that when students come into the lab, even if they've taken maybe the cell molecular foundations class, most of them simply don't have the background to start thinking of questions in cell biology yet to do their own independent research. Yeah. And so I always plug them into my research, right? I will kind of explain my project, I'll tell them what we're doing, why, and if they're interested, they're more than welcome to join. And I train them, from the get-go, and kind of so I plug them into that. Sometimes if they've been there for a couple of years, they might start asking their own questions related, and they'll kind of offshoot, but it doesn't, oftentimes they will graduate before they get there, because it's such a steep learning curve, I think. At least in, I don't know if I'm not doing it right, but that's what I feel in this field, right? I don't know if you guys can comment on that. So I don't do that very well, and I'd be actually curious to hear people's thoughts on that.
I mean, it just seems to me like some disciplines are set up really well for this kind of laboratory, group, collaborative, right? There's extra data to analyze. Some disciplines are really well set up for that. And some disciplines have this like solo, like cowboy on the lonesome prairie approach to scholarship, right? It's like you alone in the library, like meditating on classic texts, and incorporating students into that just is a challenge.
Are there ways to break it down into small parts?
I'm working on it. I am. Well, I've got English language transcripts now.
I will chime in both on your question and on Rebecca's question. I don't have a specific way to encourage people to be active in the lab or not. Actually, at the very first conversation that I have with students, my expectation number one is earnestness. You either want to work or you don't. And if you want, great, work with me. And if you don't, then it's okay, find somebody else. And I know it's a little harsh, but also there is, it's important to be productive. And if somebody doesn't want to do that, then it's perfectly fine. Like you mentioned, some people discover that they don't want to do it. And I have a very interesting example. I had freshmen who started with me, and she was very scattered, right? She would not complete assignments and had busy life, let's put it this way. And I said, well, it's okay. We can, if you are interested, we can continue that sometime later. And then 3 years later, so this was the fall semester, long time ago, right? She comes back and she says, Dr. Kestas, I want to get back into your lab. I'm ready. I say, like, what? That never happens. And she says, I worked in numerous other labs, and I think I improved myself, and I want to prove to you that I can do it. I was like, great. This is amazing. A rare case scenario. And she worked for entire half a year, and she was very productive and all that. So right now she had 2 offers of jobs, actually working with mice. And I don't know which one she chose. It's going to be either New York University or Columbia University. So a lab technician position, I'm just proud that she overcame her hesitation and unpreparedness in a way. So I think a lot of it is student driven. And in regard to your question, a similar answer, actually. I think it either happens or it doesn't. And it's in give and take with students, right? Some of them need a lot of help and you provide that, and some don't, and they just go up. And related to, also similar to what Yulia said, not everybody is contributing immediately, but there are students like that who become lab directors in a way, right? And they, during the meeting, they pop up with their own ideas and better controls and better reproducibility that even I have not thought of it. Somebody came up like, why are we evaporating with nitrogen at room temperature? If we heat up that in a water bath, the process will be, 5 times faster and we will use less nitrogen. It's like, excellent idea. I have not even thought of it. So a lot of protocols in my lab actually are student driven, but that happens only after they work like for half a year on a project and they know every in and out, right? And then you can kind of say, hey, this is boring. Why are we doing it 3 times? Here is an experiment, and I showed that doing it 2 times is sufficient. And you say, oh, good. Well, thank you for this data, right? I'll include it in my next paper. So I think that you're right, Rebecca. It depends on the field, but it also depends hugely on a student and whether they, start contributing and whether they find that niche or not, right? Michelle, you're online as well on the phone. Would you like to talk about your research? Okay.
I'm just here to listen, hear
Okay.
about all the stuff you're all doing.
All right. Thank you, Michelle.
Thanks.
As a sort of logistical question, could I ask how often do each of you get all your lab students together in like a weekly discussion meeting you have together? Like, how do you maintain those social connections and get those conversations, keep those conversations working?
Well, that could be a student question, like how often?
Yeah, this past semester, we would have weekly meetings every Friday for an hour. We'd go around and kind of give updates on what we're working on and stuff and kind of talk about what worked that week, what didn't work that week, what we need to work on the following week. And it's really nice because even though we're all working on Dr. Artemenko's research, it's very different sort of subsections of that research. So I found those weekly meetings to be very good to kind of hear what my peers were working on and how it differed from what I was working on, how it was similar to what I was working on, and kind of get new ideas and stuff like that. So that was every week.
Yeah, and we also did journal club,
Oh,
so
yeah.
students will bring in journals and just discuss journal articles and to some students, the articles that apply to their research and different techniques and new ways of studying.
Do you find that useful in the way that it helps your public speaking skills?
Yeah,
Oh, definitely.
so just
Definitely.
like journal club in general, just in other classes too. is
So
helpful.
Valeria and Quinn, is journal club or the reason you come to these weekly meetings, is that because you have a paid assistantship? Is it because it's for credit for a course, or is this purely voluntary, your own interests?
We were doing it for credit. I was doing it.
Yeah,
for
it
credit.
was for credit, but I think also voluntary interest as well, just to get the experience and to kind of learn, you know. Yeah, no, definitely. Definitely.
I
And
love
was-
journal club. I want to do journal club in anthropology.
Oh,
I need
definitely.
details.
And like Yulia said, there's kind of a big learning curve with the actual like science behind what we're doing. So journal club was really beneficial to kind of learn about those specific mechanisms and the actual scientific theory behind what we were doing. So yeah, it was very beneficial for learning in general. Yeah.
Is that like a 1 credit upper level course, or how does journal club get organized?
We have a variety of independent study ways to get independent study credits. So these guys were doing 3 credits of Bio 399, which is independent study. We also have directed research option and like a higher level independent study option, but they all essentially get, during the semester, they do it for credit. During the summer, we try to find ways for them to get paid to do that.
And those independent study courses, at least in our department, are variable in credits. And so if you have a student who doesn't have a lot of time, but has a lot of interest, you can do 1 credit hour and that person can, you know, do whatever would fit that amount of work.
Thank you.
Yeah, Liam, would you like to chime in regard of meetings, etc., and how you develop your communication skills?
So during this last summer, me and my partner of that semester, we would meet with Haddadi every week to make sure to see how we're doing for each extraction, because each total extraction took about a week. And so by the end, so once we've did all the samples, we would look at the data and be like, okay, so we have a good extraction, we have a good recovery rate, so maybe we should fine tune things, and maybe we should move to a lower concentration. Or if it's low, we'd be like, okay, so what can we do to better refine our methods to better to tune to make sure that the recovery rate is higher. And also Haddadi would also check in and she would check in while we were in the lab as well.
Yeah, so we all, many of us faculty use adaptive management technique, right? You have to move and adapt all the time. It depends on student, it depends how project goes. So Shaku, would you like to maybe chime in regard of Rebecca's question about students finding their niche or not?
I've been thinking about it. As you said, it depends on the projects that we have, and maybe considering, I try to see the strengths different students have, and if someone is, for example, good with working in the lab or bench work, I will give them more responsibilities on that side. And if someone likes to do more of the instrumentation, for example, so I try to divide the responsibilities based on their abilities so they don't get discouraged at the first place. So as they see what other people are doing, like If they watch another student using the instrument, then they are less scared to use it, so they gain more confidence. So I try to rotate it, like start with something they like and they're good at, and then move to something that they feel more comfortable to work with. I try not to impose a responsibility that the student is scared to take or doesn't feel comfortable to do. So trying, I guess what I find useful is to give this impression to the student that they are more important than the projects and they're here to learn. And if you show that they're important to you and you want to teach them something, they would want to participate. That's my experience. I don't know if I really answered your question though.
I love that. Oh, that the students are more important than the project. I mean, projects are very important in their own way, like in that moment. That's so great.
All right, and maybe I'll ask Vikrant a question in that regard, because given that he's been in my laboratory for so long and experienced so many failed projects, I think you could have a certain perspective and chime in on that. Maybe.
So in our group meetings, we usually have, well, Dr. Kestis brings up a topic that is fresh to the field and what new discoveries that are made in the field. And we talk about it, and it gets you more encouraged what contribution you can make on a personal level in the future. And it makes you more enthusiastic to work, put in more effort so that you can make some contribution in the future. So that's what I feel about it. So that's why I've been working on.
And I think we're running out of time pretty closely, so I'll give an update from today's news in science. Right, a recent paper came out in the Journal of Science demonstrating that in over 1 million people sample, right, those who were vaccinated have twofold less long COVID than those who are not vaccinated. And there have been numerous small studies about it, and when you look at small studies, they're indicative but not necessarily definitive about it. And when you see an announcement of that level, you say like, boy, that's good data, that's good data. So one of those things that you guys, you students work now in our laboratories, but you will be driving science, a generation from now, you'll be professors. So I guess our approach, many of us faculty, are pay forward, and make sure that they carry the flag, right? Okay. I guess that wraps it up, unless you want to say something, chime in.
Just to say thank you, and it was nice meeting you all again.
Thank you all for sharing. Yeah, especially the students. Thank you for coming today.
Excellent. Thank you.